Today's we are going to look at the topic called osmosis. There are various processes that takes place in living organisms, and osmosis is one of those processes that takes place in living organisms. So let us look at what osmosis is. In defining osmosis, we say that it is the process by which solvent molecules move from a region of lower concentrated solution to a region of higher concentrated solution through a semi-permeable membrane. In defining osmosis, there are some two key words that must be taken note of. These key words are solvent. And when we talk about solvent, the most common solvent is water. We can have a solvent like petrol, turpentine, kerosene, and many other ones that can serve that purpose. We are also talking about the second most important word in the definition of osmosis. That is a semi-permeable membrane. We shall later on look at what a semi-permeable membrane is. Now, these are the two key words that must be taken note of. We should also understand that in osmosis, the movement deals specifically with the solvent molecules and not solute molecules. And so, the side where the solvent is more, that becomes the lower concentrated solution. Then it directs itself to the point of a higher concentrated solution. There are some other ways by which we can define osmosis. So let us look at that. We can also define osmosis as the movement of water molecules from a weaker solution, which is more or less a higher concentration of water, to a stronger solution, that is a lower concentration of water, through a semi-permeable membrane. Here too, we take note of the two keywords, water molecules and a semi-permeable membrane. So let us move on and see. Now, the next slide that we have on the screen is showing us the process of osmosis in a chart. So let's explain how the process takes place. When you look at the diagram on the left hand with an arrow in the middle pointing towards the right hand on your screen, what is on the left hand? There is a green barrier in the beaker. That is the semi-permeable membrane. A semi-permeable membrane allows only solvent molecules to pass through it, but do not allow solute molecules or particles to pass through it. On that particular beaker, you realize that the level of water on both sides of the semi-permeable membrane are of the same height. Now, when you look at the one on the left of that, you can see only 3 molecules, solute molecules or particles, which are the red particles. On the right, you can see more than 3, more than 5 particles in there. Now, on the left side of that, you would also see that the volume of water is high, but the volume of water on the right is somehow low. So that makes the volume of water, where we have the three particles, to be noted as a lower concentrated solution. We can equally call that a weaker solution. We can also call it a dilute solution. So, most importantly, the solvent is more at that side. And where the solvent is small, that is the direction in which the movement will take place to a place where the solvent in comparison with the solute is less. So when you look at the arrow indicating the process of osmosis and we come to the diagram on the right, we realize that there is the movement of water with an arrow indicating so. As that goes on, the level of the water where we have the three particles had reduced drastically, and it has moved to the point on the right, with the level of the water rising up significantly. But take note, the three particles on the left of that still the same. On the right, the particles there are also the same. The only change that has taken place is the level of water. So it deals with the movement of solvent molecules. That is how osmosis is. Let's go to the next slide and see what is also there. We want to look at the explanation for the explanation on semi-permeable membrane. So we say that it is a material that allows solvent or water molecules to pass through it easily, but does not allow solute molecules to pass through it. That is, osmosis occurs in liquids only. So let's go to the next slide and see what is there. In the next slide, we are looking at examples of materials that can be used as a semi-permeable membrane. In looking at examples of materials that can serve as a semi-permeable membrane, we are going to look at it from two perspectives. Those that perform as a living tissue that we get as a living tissue, and those that are a non-living tissue. So when we take the living tissue, we will have plant-based tissue, whilst we have the animal-based tissues, which are used as semi-permeable membrane. So the plant tissue, we use a peeled yam, peeled cocoyam, peeled cassava, and we have many other examples that are not found or listed here. We need to know that these examples are fresh materials. Immediately you cook them, they cease to function as a semi-permeable membrane. We should know that it is the cell membranes in the cell of these materials that allows the movement of water through them. When we go back and we look at what a cell is, we will see that each cell has a cell membrane, and a cell membrane allows substances to move in and out of the cell. So basically, it is the cell membrane that is helping mainly in allowing those substances to function as a semi-permeable membrane. Let us now look at the example of living tissues from animals that can be used in the lab as a semi-permeable membrane. So we have the bladder of a pig. So when a pig is killed fresh and it is dissected, the intestines are removed. You can remove the bladder and use it for such a purpose. The skin of a toad or a frog. So here you skin the skin on its belly, and that can be used as a semi-permeable membrane. These examples I've listed are those fall under the category of living tissues. Let us now look at the next one, which is the non-living tissue. We have cellophane as the material that can be used as a semi-permeable membrane in the laboratory to demonstrate the process of osmosis. In our next slide, we are looking at factors affecting the rate of osmosis, or the conditions required for osmosis to take place. The first and foremost factor is concentration gradient. Now, the greater the difference in concentration, the faster the rate of osmosis. The greater the difference in concentration, the faster the rate of osmosis. Now, what are we saying here? We are saying that before there can be the movement of the solvent molecules from one region to the other region, it can only occur when there is a concentration gradient, or the difference in concentration with a semi-permeable membrane between these two different concentrations. As we saw in the diagram earlier, we realize that we have 3 solute particles at one end and a lot of particles at the other end, and so that brings about a difference in concentration whilst they are still in their solvent. So we saw the movement from the weaker solution to the higher concentrated solution. Let's go on. So we have the next one being the semi-permeable membrane. So for osmosis to take place, it must be separated by a semi-permeable membrane. Temperature. With respect to temperature, the higher the temperature, the faster the rate of osmosis, since kinetic energy of water molecules is increased. The water molecules need to move from one end to the other, and for them to be able to do that, they need energy to be able to move around. And for that matter, the higher the temperature, the faster the rate of osmosis. Let's just look at the next slide and see. So we want to look at examples of osmosis in plants. We need to know that these processes that takes place, which is osmosis, is taking place in living organisms, in plants, in animals. So let's look at the first example of osmosis in plants. The first one says the absorption of water from the soil by the root hairs. Now when you look at the diagram, you see spots of water there, and it is going towards the direction of the root. That is where we have the root hair. The root hair serves as a semi-permeable membrane. It allows these water molecules to enter through it, and then it indicates the process of osmosis. In another example of osmosis in plants, which is on the next slide, this has to do with the movement of water from the soil to the cell within a plant. Now, after the root hairs have absorbed the water into the plant, you can see the other arrows indicating the movement of the water upwards. And that is taking place from one cell to the other and from another cell to the other. So, the movement is taking place within a cell of a plant. And that also brings about osmosis. We are going to look at some examples of osmosis in animals. So let us look at them. The first one is the reabsorption of water in the kidney tubules in mammals. Reabsorption of water in the kidney tubules in mammals. So let me just click here. So we have the kidney, we have the unit of it called the nephron, and within the nephron we have the glomerulus. So when waste substances are generated, what happens is that they will go through it, and then the water that is going out will be reabsorbed back into the system. So let us look at the reabsorption of water into details again. So when we take the kidney is made up of a single unit called nephrons, and we have millions of it in the kidney. The kidney is responsible for removal of waste substances. So the nephrons are those that perform the function itself. And what happens is that whilst the blood is passing through those tubes in the glomerulus, they will be forced out, waste materials will be forced out, and it will pass through those convoluted tubules. Now, the body must assess itself, ensure that it has enough water in it, so it does not allow all those water to go. Now, those tubes, which are the convoluted tubules, connect to the ureter to the bladder. And so in the course of moving before they get to the ureter, the blood capillaries are surrounding those tubules, and they will reabsorb some of the water back into our system, so as there is going to create some form of what? Balance. Okay, let's go on. The next slide, we are talking about absorption of water in the colon of mammals. Yes, when we go through the process of digestion, the undigested materials would come out through the large intestine known as the colon, and eventually come out as fecal matter. In the colon, there is reabsorption, just like I explained with respect to the kidney tubules. Here too, the same happens. There are blood capillaries there. They will reabsorb some of the water back into our system, so as our system will have enough in its body. Entry of water into the cytoplasm of a body of an amoeba. That is also another example of osmosis, and this takes place in a unicellular organism like amoeba. Okay, so that is a typical example. So if you look at the cell membrane, that is the point through which these materials are able to pass through and enter into the unicellular organism. Now let us look at the differences between diffusion and osmosis. Semi-permeable membrane is required for osmosis to take place. In diffusion, there is no semi-permeable membrane that is required. In osmosis, it occurs in liquids only. In diffusion, it occurs in both liquids and gases. In osmosis, it involves the movement of only solvent particles. In diffusion, it involves the movement of any particle, either solvent or solute molecules. So take note of that differences. How do we then demonstrate the process of osmosis in a living tissue? We have made examples of materials that can be used as a semi-permeable membrane, and we made mention of the yam. So here, in an experiment to demonstrate osmosis in a living tissue, we are going to look at apparatus like yam, water trough or basin, salt, sugar, water. So procedure: we peel the yam and cut it into a cube. A hole is created in the center of the cube. Strong salt solution is then poured into it. The yam is gently placed in the basin containing the water. The setup is left for 24 hours. So let us see how that. So this is how the setup will look like with the salt solution in the. Here we have the diagram. Okay, so this is just basically an example of how the setup is going to look like. So we have the yam, the water, the beaker, the salt solution. Okay, so this is what is going to happen after 24 hours. Looking at the diagram on the left, that is the beginning of the experiment, and then those are the right, the end of the experiment. At the left of this, we have the concentrated solution in the yam cup. Look at the level of it, and then the level of the water in the beaker. But after some time, after 24 hours, look at the level of the solution in the yam cup, and then the level of solution of the water in the beaker. That should tell you that osmosis has taken place. So with that, that shows that water molecules have really moved into the cavity from the basin through the cells and the tissues of the yam. The cells and the tissues of the yam acted as a semi-permeable membrane. Let me be quick here to give some explanation. Now, the explanation I am giving here is that when it comes to osmosis, as a student, you should always look out for, when a practical question is asked, you should always look out for the place where we have the highly concentrated solution, and then we have the dilute solution or the weaker solution or whatsoever. And when you are able to identify that, we should know that the movement will take place from the weaker solution to the stronger solution. When you have that at the back of your mind, and then you have a semi-permeable membrane which is functional, then you'll be able to easily predict where the movement will take place and how the levels are going to be. So that is what it is. Take note of that anytime you meet any question. So the diagram that we just saw, the solution can be in any, either in the beaker or there, or in the yam cup, and you just have to just know which one is highly concentrated or dilute, and then you trace the path and move on. So here we have another kind of setup showing the Tistel funnel. Okay, and here we use it for those other. It's also used in this. So we have the semi-permeable membrane, as you can see. You have the sugar solution and all those things. So the semi-permeable membrane here, where we are using the Tistel funnel, could be the cellophane, could be the skin of a toad, could be the bladder of a pig. So this setup is just to show you that you can also meet a question like that. And so when you meet it, don't be taken aback. Let's look at the very final aspect for us to end this lesson. So we have two diagrams here. Here they are using the cellophane as the semi-permeable membrane. So you can see on both sides, on the left hand side, the A, you saw the initial level of the solution, and then it rises up. So that shows that when we use this as the material for the experiment, that is the expected results. The initial level at the beginning, and then the final level at that end. Then we come to the B. The B, there is water in the Tistel funnel, water in the cellophane, in the beaker. So at the end of the day, the level remains unchanged. We made mention of the fact that for the process to take place, a concentration gradient must exist. And so the concentration gradient has been there. The concentration gradient has been the same, and therefore there is no movement from one end to the other. So we're going to end this lesson by saying that we should remember the definition of osmosis, the factors that affect the rate of osmosis, examples of materials that can be used in making serving as osmosis, and then the experiment that we have just looked at. Thank you very much, and watch out for the video on this practical example. Thank you, and have a nice day.